Hi,
Thank
so
you.
I'm here with Rob Wust. Is that how you pronounce it? Wust?
Wust, yes, with a Larry
So
U.
Wust. Okay, Rob Wust.
Yeah,
Sorry about
yeah.
that. To discuss, well, one it was his new study on the impacts on muscle of an exercise challenge that caused a lot of stir when it was published earlier this month. So, Rob, why don't we first why don't you just lay out for us what it was that you studied and what you were trying to look at?
Yeah. So, thanks for the invitation, Italy. Very nice to be able to talk to you about this study. So we got a request about 3 years ago from a clinician in the local hospital who said she was seeing long COVID patients, and she was obviously there was obviously something wrong with these people because they couldn't do exercise, they'd actually worsen all the symptoms after exercise. They got really fatigued, and she was convinced that there was something wrong with these people.
She didn't think
Inside
it was
the skeletal muscle.
psychological or that they were depressed or that there was a psychological
Exactly.
social.
Exactly. That was the initial, of course, theory that maybe there is a burnout. They have something psychological, and therefore they should just get exercising again, and then they should probably get better. Now, the idea that was not the case. She really saw these people, and she was like, well, before the COVID infection, they were actually pretty okay. So why would this cannot be true? Let's look inside the skeletal muscles. And I'm a skeletal muscle physiologist or exercise physiologist at the local hospital of the local university. And we do a lot of work with inactivity and different diseases and heart failure, diabetes, etc. Trying to understand what are the underlying mechanisms that are causing the reduced exercise tolerance. So I thought, well, this is a perfect opportunity to look at this together.
And did you
So
know about ME at that time or MECFS, or was it something that was on your radar? Or post-exertional malaise, was that something that was on your radar?
it was not on my radar at all. I had to admit that I didn't know anything about ME. I didn't know anything about post-exertional malaise. I only knew that these people there was something wrong with these people that likely resulted in some skeletal muscle changes that could underlie these increased levels of fatigue. After this initial conversation, I started digging into this, and we heard about its two-day exercise tests to diagnose post-exertional malaise. And I thought, well, actually, if we want to do better than this, rather than doing a 2-day exercise test to diagnose PEM, so to say, why don't we use the knowledge from those exercise tests and apply it to skeletal muscle tests? So instead of a 2-day exercise test that suggested that there is a peripheral limitation to exercise or a peripheral component to the post-exertional malaise, let's look then inside the skeletal muscle. Let's do a 2-day biopsy test. So we wrote the ethical approval, and we just thought, okay, let's do an exercise test, an exercise challenge to measure the exercise capacity.
So basically, the biopsy sort of substituted for the second CPET test because you were measuring the impacts of the first one rather than by doing a second CPAP, but just by looking at the tissue.
Exactly. Yes, exactly. So we did really focus on the skeletal muscle tissue because that really helps us to understand better what happens upon exercise inside the skeletal muscle tissue that can explain maybe the feelings of additional fatigue. So we set out this sort of cross-sectional comparison as well as a longitudinal comparison, what the acute effects are of exercise on patients, control patients, healthy people, and people with long COVID.
So longitudinal in the sense that you tested them at baseline and then you tested them again after the challenge.
1 day after a maximal exercise test. So we use a maximum exercise test for 2 reasons: to test their exercise capacity as well as inducing PEM. So we were sure, because it's maximum exercise, we were sure to go over the threshold where post-exertional malaise is being induced, and we asked them back the next day. So all our
how did you know that when you say your intention was to induce PEM? How did you know that what you were inducing was PEM as opposed to just normal, responses to exercise. I mean, what
Yeah.
are you defining PEM? The patients already came in with a defined PEM?
Yes, exactly. So we specifically selected participants who were positive on the DSQ PEM questionnaire. So they had to have suffered from post-exertional malaise or PEM. And they also had to undergo a mild initial infection, no hospitalization, and no other comorbidities that can explain possibly levels of fatigue or additional fatigue. So we really screened patients where we were certain that we had a relatively homogeneous population of patients,
And
but they had to PEM. Yeah.
one of the things with PEM is that it's often a delayed, by 24 hours or 48 hours. Was that distinguished in the selection of the patients who had PEM, or was it, simply self-reported that they had PEM, but not identified very much exactly what PEM was?
Yeah, so indeed, there is not really a very clear description of what, or a quantification of PEM in that sense. So some patients have it already after a few hours, some are already acutely after exercise, others have a bit of delayed onset after 1 or 2 days. So we actually had a big discussion about these things. When is the best time to do the second biopsy, so to say, after the initial exercise test? Is it 24 or 48 hours? And what we then ended up doing is the 24 hours, because then we were certain that the time was short enough after the exercise test. So if the exercise test did something, you would see it already after 24 hours, rather than 48 hours, where maybe other things. Well, it could have been the top, of course, of the PEM. We don't know because we don't ask that. We didn't ask them the day after again.
Right.
Also, logistically, it turned out to be easier to plan this, to plan all the exercise tests on one particular day and all the biopsy tests on another day. So it was logistically easier to do one day after exercise.
Okay, so what were and you compared them to long COVID patients, no, to COVID patients who had recovered fully and were back to normal.
Exactly.
Okay.
Exactly. So we used a healthy control population. Generally, they were relatives of the patients because they knew what a disease entitled and they really wanted to contribute to science. So we have very similar demographics of the control group. And yes, all the control people also had to have had acute COVID but recovered properly.
So what were your findings then?
What are the findings exactly? So we divided upon two aspects. So we found structural changes in the skeletal muscle that can predispose tissue to be more fatigable. So muscle consists of red and white muscles, and we saw more white type muscles in the patients with long COVID.
At baseline.
At baseline, exactly. At baseline. Also their energy production capacities, so to say, so the organelles that produce energy or mitochondria, they are key to produce enough ATP. And those were all that the amount of the maximal capacity for these mitochondria to produce energy was also reduced. Now we know from lots of other diseases that those two things together, they can predispose people to become more fatigable. So that's what we found. So that the general feelings of fatigue can be partly explained by something inside the skeletal muscle. It doesn't mean that everything is explained by it, but at least there is a contribution to it. Now for the longitudinal comparison, the before and after the exercise, we looked for a specific marker that is able to quickly change upon exercise, because your muscle contractile material, so the white or the red muscles, do not change very quickly, so that doesn't contribute to the feeling or the development of post-exertional malaise. So we looked for rather quick changes, and that's what we found in the metabolism. So these mitochondria, these energy-producing organelles, they're the ones that are that reduce upon in function upon exercise challenge, And that can essentially lead to a sort of vicious circle. If you keep going into PEM, you get this vicious circle of reducing your ATP producing organelles, and that can lead to a vicious circle of symptoms of fatigue. So that was one of the things that we spotted. We also looked for this microclot theory. So the microclot theory in the field,
The amyloid microclots.
the amyloid microclots, exactly, the amyloid deposits, or microclots called in easy layman's terms. So we did see changes or differences between groups before the exercise challenge, but we don't really know what they do inside the skeletal muscle tissue. So they are there, but the idea was, or still is from some groups, is that they can block capillary. So they can block blood flow and cause local hypoxia that can explain the mitochondrial differences of fatigue. So this was going to be a super.
You found them in tissue as opposed to in the capillaries.
Exactly. So they were not in the capillaries. So we thought it was going to be a super easy, straightforward project where the clots are going to be in the capillaries. End of the story, because it's just everything is related to hypoxia. We didn't find them there. We only found them outside the tissue or outside the capillaries, in between the muscle fibers. And we simply don't know what they do there or what they cause. But we do know now that they also increase in concentration or in density upon an exercise challenge with the induction of PEM. Whether they are there and they cause any feelings of pain or fatigue, we simply have no clue, because we don't know what they feel like. These microclots, if they can.
So is that research compatible with the research finding them in in capillaries, or how do you square those two sets of researches?
Yeah, so we of course already quite early on in the process spoke with the group in South Africa who developed this theory, and the group of Vrije Pretorius now is actually analyzing all our blood measurements so that we can also sort of put together these muscle changes as well as the blood changes, and maybe have an idea of where they are produced, these clots, and where they're moving to. We don't know. So
basically, you're doing some little bit of collaboration with the other group to try to figure out what's going on, and there's some way to square these findings with each other.
Exactly, that's indeed the case. I mean, the group in South Africa suggested that there is there are autoantibodies inside these clots, and of course that would be in line with the idea that it's becoming an autoimmune disease. But then the question is, what are these autoantibodies doing inside skeletal muscle? We don't know.
No, is your idea that I mean, you were looking at leg muscle biopsies in one of the leg muscles. Was your idea that the effects were more pronounced there because those were the muscles being used in the cycle challenge, or is your sense that this is happening in the muscles all over the body, but you just tested it in one muscle? So, I mean, is this a systemic reaction, do you think, or is this a localized reaction?
Well, in terms of the metabolic change that we see, we do think that they are rather local. I mean, the muscle needs to be activated to have an effect. But we don't know for certain, because we also know, for instance, that likely there's a factor in the bloodstream that causes other symptoms to contribute to other symptoms. And if it's in the bloodstream, it can exert other effects in other tissues as well. We mainly focus on the skeletal muscle tissue because if there is a difference, the first place where we should look is in the skeletal muscle and not in an arm muscle or so. But we don't know really whether the arm muscles are affected in a similar way.
Even if they're just doing a leg exercise.
Yeah, if you do leg exercise, whether your arm muscles also change. The fact, I mean, there are some studies that show this more long distance effect, so to say, from muscle to muscle, but they are very difficult to spot. So I have the feeling that there will be too much variability, and the effects are too small to also really affect upper arm muscles, for instance.
So, what about mental, PEM from mental exertion? How does that? I mean, that was one of the things that I was sort of reading on, online and different comments, and that was one of the questions that was raised. How does, I mean, the CPET studies also obviously look at physical provocation of PEM, but how does this square with mental
Yeah.
provocation of
Yeah.
post-exertional malaise?
Yeah, yeah, that's a super good question. I simply don't know the answer yet. What we do with our studies, because I'm a muscle physiologist, I focus on exercise and muscle function. So we really use the physical exertion as a way to induce PEM. Now, I know that also, cognitive exertion can induce PEM, but okay, but I need to explain a little bit more detail, the reason of thinking in this way. If physical activity causes skeletal muscle changes, we also, of course, saw mental or cognitive changes in the patients that we saw as part of the study. Brain fog, concentration problems, sleep problems, etc., etc.
Did
So
those
we
also
do
function as part of the PEM, or were they just
exactly. So as part of the exercise challenge, we also see neurological
right?
changes, so to say, but we only focus that on in terms of symptom description. So we do think that there is a crossover, so to say, to other symptoms as well, not only skeletal muscle changes. We focus particularly on the skeletal muscle in our study. If cognitive changes, cognitive exertion is, of course, can likely induce cognitive PEM. Whether that also causes skeletal muscle PEM, we don't know.
I
Whether the
see.
muscle abnormalities are worsened with cognitive PEM, we don't know. But it's likely that the cognitive PEM induction, so to say, cognitive challenge induces cognitive type of neurological changes in the brain.
So you're
But
positing that
actually,
there may be separate there may be separate kinds of PEM, or that they have somewhat different mechanisms, whether it's a mental exertion or a physical exertion, but they're leading to a similar, but not necessarily exactly the same state of post-exertional malaise.
yeah, it could be, but it's indeed there could be two types of PEM, so to say, or two types of flavors of PEM. And actually, a Dutch group now is studying this in more detail. So they do brain scans before and after a cognitive challenge to
Right.
really see what happens in the brain with the cognitive challenge. And of course, we are working together to see whether we can also do whether we can understand the problem from different perspectives, essentially. Yeah.
So you got some responses on Twitter, a couple
Yeah.
from Professor Carson, um, who was polite, but made a couple of points, which I also wrote about in my blog post about it. The first one was, what was his first point about? Um, no, I can't remember what his points were.
No, one of the comments were related to inactivity, deconditioning of patients.
Right, right.
Uh,
So one of their, their hypothesis is that a lot of this is due to deconditioning, and that's all the effect. So that was one of his comments, and the other comment I can't remember, but also I think was equally. Whether this was how do you separate this from just DOMS, from just basically muscle soreness after exercise, which we all sort of have. So those were the two things that he pushed back. Do you want to sort of respond to those?
Yeah, of course. So the deconditioning one is a relatively simple one to dismiss because we know also from the work from our lab. I mean, I'm a muscle physiologist, and we've worked for the last 5 or 6 years on a big bed rest project in collaboration with ESA and NASA and the space agencies to really understand the effects of bed rest on skeletal muscle changes. Now, for this study that actually also came out about 2 weeks ago now,
Which was coincidence, right? I mean, it
super
wasn't
coincidence.
ME
Super
study.
coincidence.
It was unrelated.
Exactly. Completely unrelated topic. Well, not a completely unrelated paper, but it was actually accepted within 2 days from each other. So we worked on those two projects for a long time. And in there, we had healthy people undergoing 60 days of bed rest. And we took muscle biopsies at the start and at the end.
Question about this.
And
How did you find 60 healthy people who were willing to be
yeah,
put in severe bed rest for 2 months. I mean, who has a life like that?
Yeah, so there were 24 people, that 24 healthy volunteers. They sign up because they're interested in space research, and they also get funding, of course, for this. They get paid to do this, but mainly it's for their there is a very strict psychological and medical questionnaires and essays they need to go through before they can be admitted to the study, to make sure that there's not a real big issue with health, for instance, health concerns. Now, those people that are just being inactive, they don't develop any signs of post-exertional malaise. So they're healthy people. If they go out of bed, of course they have problems with standing up, as what also happens in space. So there is some orthostatic intolerance that we also see in patients. That's actually quite interesting, but we don't see any signs of, if they start exercising again, that their symptoms worsens, because they don't have any symptoms of brain fog or POTS, or the orthostatic intolerance. So we know that these people, if they start exercising again, they are returning back to their pre bed rest values in terms
Right, so
of
they're abnormal deconditioned people. There's nothing wrong with them, but as soon as they start doing things again, then they get reconditioned.
Exactly. And also the skeletal muscle abnormality, not abnormalities, but the changes that we see are completely different. There's a lot of muscle atrophy occurring in those groups. The reduction in capillary density is higher. So that's what we did not see, for instance, in the patients.
Now, don't we already know that? Didn't we already know that? I mean, why do we need a new bed rest study to sort of show that this isn't deconditioning? It sort of seems sort of silly to even have to bring that up.
Yeah, that's true. So we know already from the 60s and the 70s, from the last century, that what effects are of bed rest on skeletal muscle changes, and how recovery improves that again. But it's very simple to just dismiss these changes that we see as being inactive. I mean, the physical activity levels of the patients were a little bit less, but they were not completely bedridden. Fiber type shifts, for instance, that we see in the patients does not occur with bed rest. So we know that there are some specific features in the skeletal muscle that we don't see in the bed rest studies, but we do see in the patients.
And so how about the other concern raised by Professor Carson, which was about how is this just different from the mechanism of, people after exercise get weaker, or have, there's exercising capacity that happens after that.
Yeah, that's of course. I mean,
And you
we do
essentially controlled for that, and I mean that was basically your comparison group, from what I would gather. That's how you showed that wasn't the case.
Yeah, so we do of course have a control group that also underwent the same exercise, and we also tested it for all the same things, and we did see some muscle damage markers in the controls as well, which is consistent with the idea that there is indeed some sort of damage happening after physical activity. But yeah, there was more in the patients, so it was what was actually worse. It was different. It was more obvious. The other thing that we noticed is that when we took out, the two other things that are worth adding here is that before the exercise challenge started, we took a biopsy. There we also spotted in some participants signs of necrosis and regeneration, suggesting of previous or past problems. So we do also see, not only after the exercise challenge, but also before, some signs that there is something wrong in the skeletal muscle in terms of their abnormalities in terms of regeneration and function, so structure.
But you can see, what you're saying is that when you looked at them before the exercise challenge, you already saw some signs that were different, that indicated previous damage along these lines, previous muscle necrosis, was it? Or?
Yes, well, we of course the problem is a little bit with biopsy studies is that the biopsy is as small as a grain of rice.
Mm-hmm.
So in some people we have 100 cells, in other people we have 200 cells we can look at. So it's always a risk that we don't see something because we just don't have enough cells to look at.
Mhm.
So it's always a percentage, so to say, of cells that give an answer essentially to that shows something different. So it's also a matter of chance and to see the muscle damage markers in that particular biopsy, depending on the size.
I see.
So there are some technical problems that we cannot solve essentially. But the only option is to take a bigger piece of muscle, but that's becoming unethical. So yes, we have to, but we are looking for various ways to the tissue, and also what they look like, what a tissue looks like from when we take it out of when we take the biopsy out. We sometimes already spotted some abnormalities, by looking at them and looking at the tissue.
So, how do you respond when you get this kind of pushback that you think that seems not really relevant to the actual study, or seems sort of, not quite responsive to the study itself?
Yeah, it's of course sometimes they are, criticism is normal. I mean, there's a Dutch saying big trees get a lot of wind. It's because they get a lot of attention.
Yeah.
So there is, of course, a lot of, when you get a lot of attention, you get a lot of comments and people talk about it. So, I mean, that's something normal in the scientific field, and that's, that is new for me, so to say, because it's the first study that gets so much attention. But you try to be, polite, because in the end they are colleagues. In some cases, you just don't reply because it's it relates to vaccination status. That does have nothing to do with the study design or the study outcome. So you do try to, of course, we try to improve things. We've been thinking about all types of criticism for more than a year before publishing this. So this is not something that goes overnight into this journal. So we are doing all the work in the lab to rule out any other potential technical or logistical flaws to make sure that what we present is really obvious in the from the data.
So two questions. One, and really one question, I guess. What's next with this line of research, and also how does this relate? What are you doing in terms of trying to bring this into relation with ME or MECFS, whatever we're calling it, ME? Let's call it that. To bring this into these findings, to examine, is this also going on with ME, which maybe it very well might be.
Yeah, so this actually was an interesting thing, because we actually started with the idea, let's do 5 people and let's just see what happens. Now this went a little bit out of hand, so to say, and we actually went for the in scientific terms, for the low-hanging fruit. We only tested the most obvious things, and we tried to make a story about this. But we haven't dived deeper into the skeletal muscle abnormalities that we see. We can follow up on all the different aspects of the study. To really dive deeper into the mechanisms behind there, or the X factor that cause all these changes. So yes, that's one of the pathways.
So these you found associations, but we haven't identified what's causal or what's causing what.
Exactly.
Basically. So you
exactly. So we do see variability between patients in terms of mitochondrial function, in terms of fiber type distribution, etc. But their fatigue scores were just really black and white. So we do know that all patients develop PEM, but some actually were ex-bodybuilders. So they're also bodybuilders. Yeah. So there are people in the cohort that actually had big fibers. They actually were able to push on a bike relatively okay, but still they develop PEM day after. So there's definitely something that is causing that, and that's what we're looking for. So that's one aspect. Of course, we because of the symptom of post-exertional malaise being also prevalent in the ME population, we now test also ME patients, because that's an obvious next step to do, to see whether the same pathway, the same
Mechanisms.
symptoms also occurs in another patient population that has the same symptoms, because then it makes the story stronger that there is something contributing to this. So those are two sort of larger things to dive into. And of course, I mean, we're looking for ways to diagnose the post-exertional malaise. Can we somehow, find a threshold of PEM? We know that in some patients the threshold is relatively high, that are probably also not so severely ill. But the ones that have a very low threshold, those are the ones who develop PEM if they come out of bed,
Right.
or if they walk out of the house. And that's what also, how can we diagnose this, and can we make sure that threshold for PEM induction is higher?
So basically, you want to find some sort of marker that you can use from these. And what about any possible? I mean, how does this play into any hope for treatment for patients at this point?
Yeah, because I think there are two comments to be made about this. So we don't know the exact X factor that causes these changes. So of course we have some ideas, and I think the next year we will try out some of those things about this. What these X factors may be. I mean, there is of course a link with autoimmunity. So I mean, you can try something with that. But the other one is to do symptomology treatment, so to say, so where we can maybe increase the threshold for PEM or shorten the duration where PEM is occurring.
Mm-hmm.
And that's also something that we're looking for. Maybe that doesn't solve the problem, but at least hires the amount of activity people can do on a daily basis without going into PEM.
So just as a last question, what do you say your findings? I mean, there's obviously sort of a split between those who think these issues are related to, quote, psychosocial strain or, PTSD from the pandemic or anxiety, depression, all the other things, deconditioning. What does your research say about, have to say about that kind of theory related to these deficits that people are experiencing and reporting?
I think that in general, all the long COVID patients that we see really want to work again, want to go back to having a healthy life, a social life, and I think it's clear from the patient interaction that we've had that that's something that is missing, and they really want to go back, but they just cannot. So just a bit of recognition that there is really something going on with these patients, I think, is important. And that it's not just simple anxiety or deconditioning that causes all these problems, but there is something more underlying these problems that deserve attention, and that it's not a simple solution either. With try to sleep better, or try to exercise a bit more, or maybe you need to change your diet. We know that doesn't treat patients in that way.
Well, let's just say those things may help around the margins to some extent.
Exactly, there are some that you can within certain. Of course, we don't say that patients should not exercise. Patients should exercise below the limits where PEM induction is occurring. So we do, of course, suggest patients to remain active within their boundaries. I think that's super important to highlight. But that doesn't solve the problem,
right,
that these patients have. So then there's more needed to do that.
Okay, Rob, thanks very much. I appreciate it. It's a few minutes till your time there, so I'm going to stop the recording right now.